So does everybody see the slides?
Yes.
Okay,
Yes.
perfect. Excellent. So first of all, thank you very much for the invitation and for the opportunity for me to tell you about the gene therapy that we're developing at Youthereum. It's my favorite topic, talking about epigenetic rejuvenation. Also, we call it sometimes epigenetic rollback, because we are rolling back the epigenetics to a more younger state with partial reprogramming, which is using Yamanaka factors, OSKM factors, for partial reprogramming, partially rolling back the epigenetics to what we at least hope would be a younger state. So just very briefly, this is mainly a scientific presentation, but just a little bit of background of what Youthereum is. It's a company I founded in 2017 with the goal, as I mentioned already, to translate this paradigm of epigenetic rejuvenation or partial reprogramming into a therapy. And we envision it being used as a gene therapy. Other teams, other companies right now are using different approaches, but we think gene therapy is the most optimal way to use this partial reprogramming on a regular basis with periodic epigenetic rollbacks with the goal of rejuvenating the organism and at least freezing our mortality rate at a certain point and stopping it from exponentially increasing as it normally does for human beings or other mammals as well. And I'll talk more about this during this presentation. We're a very small startup. We have a minimalist philosophy. We try to avoid any kind of overhead or expenses, and all the funding we receive, it's mostly donations or just my own money right now. Right now, it's all going for research. There's no other costs, but we'll be raising more funding for to be able to do this full time, and we'll be, of course, spending a little bit on operations, on burn rate, but still, most of the money will be going towards research and turning this paradigm into a therapy. These are the current team members. It's just the three of us. Initially, there were 6 people in 2017, but some people have started their own startups and they're doing this exclusively now. And at this point, there's just 2 other people who are volunteering their time and expertise to the project. But we have a lot of advisors. So at this point, they outnumber the team members 2 to 1. I'm sure many of these names are familiar to you, especially Aubrey DeGray. I think everybody knows Aubrey. And there are other advisors who are helping us with their scientific expertise. So as I mentioned, our philosophy is to translate this paradigm as soon as possible. And our paradigm of operations is mission first, because we want to see rejuvenation developed as a therapy by anybody, not necessarily by us, because I think we have all common goal, all of humanity, or all the movement for defeating aging. The most important thing is that we develop something as quick as possible. It doesn't have to be this paradigm. It has to be any sort of therapy that can provide us and other people with some measurable life extension, because right now there's nothing that is able to extend human life, unfortunately, by any significant margin. So we don't necessarily need ourselves to succeed. We want somebody to succeed. So we're not very traditional in that sense, that like a traditional biotech company, that we view others as competitors. We actually want somebody to succeed. If it's a competitor, if it's other company developing even the same approach, we'll be more than happy, because number one goal is not to make money, but to create a therapy that can rejuvenate people. If it's us, okay, great. We're trying to do our part, because actually that's why I started Youthereum in 2017, because I didn't see anybody else translating this paradigm. This paradigm to me was like a bombshell going off, the partial reprogramming and epigenetic rejuvenation, that I thought, in a few months there should be 10 companies trying to translate it, but nobody was. And for a year, Youthereum was the only company trying to translate it. But in any case, and I think for this to succeed, we need to be as open as possible with our data, with our research, with our results. And this is what we're doing. We're sharing our progress online. And I hope our colleagues also do this in a more open way, because I think traditional way of doing science is a little bit counterproductive to actually the main problem we're facing, which is defeating aging, and not being afraid of being scooped by our colleagues or having the best paper out there. But this is a different topic that I'm also sometimes speaking about, this kind of problems with the scientific academia structure that is inhibiting, I think, the progress of rejuvenation research. But today I won't talk about it anymore. So let's dive into the science, which is, I think it's fascinating. And I wanted, I can talk for hours, but today Natalia said I only have, I think, another 30 minutes remaining. I'll try to be quick. The paradigm of epigenetic rejuvenation. This slide just tries to very concisely sum it up, what it is, and then I'll go into details. So what it is, it's the epigenetic mechanisms that control all kinds of gene expression in our organism. I think they're largely responsible for aging as well. And I'll be presenting evidence why I think that is in later slides, but here it's just a summary that, first of all, we've been able to epigenetically roll back cells using Yamanaka factors, not we, but I mean we as a scientific community, which was the first clue that rejuvenation is possible and epigenetic rejuvenation is possible, because this happens epigenetically. You take an old cell, use Yamanaka factors, it epigenetically is reset to a pluripotent cell, and it also ameliorates all hallmarks of aging. Also, another point is that we actually discovered that there are epigenetic clocks in mammals and humans, and this is also, I think, more evidence that there's a huge component, if not the primary component, of epigenetics controlling aging. And of course, the most important study that impressed me so much that I started Youthereum was the 2016 study by Ocampo and others from the Salk Institute, which used partial reprogramming in vivo, in a live organism, to greatly extend the lifespan of those mice, and also show that it is possible to rejuvenate already older animals. So with that said, let's go into more details about everything, all the aspects that I mentioned in that slide. And the first important question, which I think is very important to answer, is why do we age? Of course, the obvious answer, which you can always answer in biology, because that's what benefits the genes, that's most profitable for the genes to do. They want a certain lifespan for a certain ecological niche. That's why different species have very differing lifespans. And sometimes people think that there's only a small percentage of lifespan that is determined genetically. They sometimes say that there are studies that show that only 10% of our lifespan depends on the genes, or 20% of our lifespan depends on genes. And that is true between individuals of the same species, that, there's a lot more of environmental impact for, say, different people, why someone lives to 100 and another lives to 60. Yes, within species, the genes that differ in those two individuals have very little impact on why, they will live, how long they live. But between species, this is 100% genetically determined. Lifespan is a genetically determined trait of a species. There's a wide range usually, but that range is determined. Say, between the mouse lives between 2.5 and 3 years, but no matter what you do, it won't live for 5 years. It certainly won't live for 50 years. Like say bats. Bats are also mammals. They live up to 40 years old, and most of them live 20 to 30 years old. And that is also genetically determined. So with that, the great example, of course, are turtles or Greenland sharks, whales, which live for hundreds of years. And no human has ever gotten even close to that number. Jean Calment was the oldest record holder, 122 years, which, there's some dispute, or there was at least a couple of years back, but it's still, humans are nowhere near close to the limit of a lifespan that other organisms can reach. And this is very encouraging, because it shows that there's no fundamental reason in biology why we couldn't live for hundreds of years, because there are animals, even mammals, that live 200 years. So at least for us, the bar is set much higher than right now. Also, we know that, as I mentioned, lifespan is genetically determined, and lifespan varies widely between different species, which, at least to me, tells me that this is a genetic adaptation. A given ecological niche for a given species is genetically determined for an optimal lifespan. There's some ecological niches for mice where it's optimal for the genes to very quickly have a turnover. That's why mice live for 2 years, 3 years. But at the same time, naked mole rat, there's a different ecological niche. They live for up to 30 years, or 35, I think is the record right now. But they're very, genetically similar. They haven't. They're pretty close relatives, evolutionarily speaking. But we understand that this huge variation is genetically determined and has been an evolved trait. And this is also good news because we understand that there's some plasticity in lifespan that we can play with, even with very close genetically related species, which for us, for humans, I think is encouraging because we can, without greatly modifying our genes, we can hope to be able to increase our lifespan significantly. Another evidence of that lifespan is a genetically determined trait is that, as I mentioned, closely related species can have very different lifespan. And the best example of this that I know is the perch genus. There's different species of perch, like the genus is very closely related different species, like Homo, like people. There's, the Neanderthals, the Denisovans, they're all people, they're all humans, they can all interbreed, but within the same genus of perch species, some fish live for 12 years, and others live for 200 years. So can you imagine like different species of humans having such a huge variation in lifespan? To me, it says that this is, again, a genetically evolved trait, evolutionarily involved trait that is genetically determined. And it's not just, a couple of different species in that genus. There's 50 different species of perch with a whole gamut, very smooth range of differing lifespans. So this tells me that it is evolutionarily very strongly determined by genes. And another evidence of this that I see is in the lab, that without genetic manipulation, nobody has succeeded in greatly extending lifespan. Yes, we've used, metformin, rapamycin, and other geoprotectors to increase lifespan by 10, 15, 20%. In mice, still the best therapy to extend lifespan is caloric restriction, starving. There's no other geoprotector that can, unfortunately, yet beat that. But with genetic manipulations, we've been able to extend the lifespan of C. elegans by 10 times, just with one knockout of one gene, and 2 gene knockout 5 times. And to me, in mice, so of course the insulin growth factor knockout, they live up to almost twice as much. And to me, this says that, again, genes are the primary regulator of lifespan. And of course, the loudest evidence of this that I see in nature is social insects, where the same DNA, identical DNA between, 2 twins can be either result in a huge lifespan for like the queen and a very short lifespan for worker castes. Like in ants, in bees, the workers live for months, queens live for years. The best example is, not the best, one of the examples is the honeybee, where, they're very similar looking body composition. There's no huge difference, but the queen lives for 8 years, up to 8 years, and the workers live for, even just in the summer, they live for weeks, less than 2 months. And obviously they have the same DNA. And some people say that maybe workers live so short because they work very hard, they have to fly and forage for food, and that takes a toll on them. But those people seem to forget that queens, they are experiencing huge levels of stress because they reproduce in huge quantities. Like they can lay up to 2,000 eggs per day at maximum intervals, which is more than their weight in eggs. Can you imagine like a woman having 2,000 children per day? Of course, they're undeveloped eggs, but still, it takes a lot of physical toll on or not toll, it has a physical activity to do this kind of work for a queen, but still they can live for 8 years. So obviously it's not wear and tear that causes them to live in such a very different duration between the castes. Same thing we observe with ants, except the range is even greater. The queen ant in black garden ants can live up to 30 years. That small, very little insect can live for 30 years. Obviously, this is not some wear and tear to me that for some reason limits the worker ants to live for only 30 for only 1 or 2 years. And the biggest evidence of the plasticity of this and that it's epigenetically controlled, I see in the Indian jumping ant, where actually what can happen is that the worker ant, in case the queen dies, can become a queen, can become a reproductive ant. Actually, it's called gamma gates, but don't worry about it. And not only when it does that, it starts being a reproductive species, individual, but it also greatly extends its lifespan, which again, this happens epigenetically. This reprogramming process of a worker reprogramming to be a queen happens epigenetically. And this greatly increases its lifespan, which to me is all the more evidence that there is inherent limitation, epigenetic limitation in different species, that genes set for a precise lifespan. And obviously, the genes that give the gift of long life to a queen, they're present in the workers, but for some reason they're silenced. They're not being used in a worker ant or worker bee, except when all of a sudden this worker needs to turn into a queen. Then those genes become activated and are able to prolong lifespan. So to me, these are all clues that aging is not just epigenetically determined, it's programmed into the genes, and then it's executed. The program of life history or ontogenesis is executed epigenetically. Another example, forget about social insects, because sometimes you can view different castes as different like DNA programs that are kind of packaged into the same DNA, but they're very different depending on the path that in the childhood the ant or the bee takes. And they can view them as two different DNA kind of joined together. And the genetic program for a queen is very different from a genetic program for a worker. But at the same time, of course, the genes that are present and are able to give the queen such a long life are present in a worker. But even so, let's forget about the social animals and look at the different animals where there's no different castes. There's just one type of individual which can lead very different life. I mean, the lifespan can be very different depending on the time where it's born, on the conditions. And monarch butterfly is a great example, because most of the generations of monarch butterfly, they live in the United States, they only live for 1 or 2 months. But the last generation, the generation that needs to overwinter and that flies down to Mexico to do so, can live up to 9 months. It's the same DNA, same individual, but for when the genes needed to continue reproducing, they allow it to live 9 times longer. And it's it's a demanding process to fly down to Mexico, overwinter there, get pregnant because they actually reproduce in Mexico as well, and then fly back. So it speaks against the kind of the wear and tear view of what limits lifespan. And to me, speaks volumes about the epigenetic control of lifespan, that genes either allow us as their copying machines to live long, or they don't. And even in mammals, we see this. There's this montane vole in the States, in Montana, this little vole that lives, has a different lifespan depending on the time of the year when it's born. They live very short, they live for less than a year. And if they're born in the spring or the summer, they very quickly become sexually mature. Within 3 weeks after birth, they can be sexually mature. But if they're born in the fall, they actually delay their puberty. They delay it until this next spring, which is a 7-time difference. And can you imagine that children weren't sexually mature at like 12 years old, and instead for some reason they delayed it until they're 80 years old. And then when they're 80, they become adults, and they just keep being children for all these years. Well, these mammals can do this, and of course they do so epigenetically, because the epigenetic program for puberty is just not turned on. It's being put on pause. And this, to me, more evidence that there's so much plasticity in our organism with our genetic programs that are epigenetically executed, that we can hope that aging is also, we would be able to find epigenetic control levers for the humans, not just for organisms that are already have inherently these mechanisms. But for humans, I think, which we can adjust for our own, not for the genes benefits, but for our own benefits to prolong our lifespan. So this is just a summary slide of what the previous observations tell me, and they tell me that aging is driven by epigenetics. Epigenetic controls all stages of organismal development, which, it's pretty obvious. Epigenetics is just a differential control of gene expressions. Some genes need to be on at different times. Other genes that need to be, very different sets of genes need to be on in different sets of tissues. Your brain cell needs to be expressing different genes from your skin cell, and different stages of development require different, levels of genes being turned on, or different genes being turned on. It's probably, and it has to be synchronized. Obviously, it has to be synchronized so that, your liver doesn't grow bigger than your body because it started puberty prematurely. There's obviously a synchronization center somewhere. Most likely it's in the brain. Most likely it's in the hypothalamus, the pituitary gland. That's the HP star axis, which controls different stages of development and synchronizes them, not just controls it, but synchronizes them. And epigenetics in each of the cell is the next level of synchronization, to my mind. Basically, it takes cues from the probably hormonal regulation. That's how puberty starts, right? Your hormonal levels change, and they turn in different tissues, which at the next level in different cells, turn on different programs, which start, again, proliferation, or you start growing different organs, or at the same time, for example, when menopause starts, that's another change in the hormonal levels and everything. So there's different levels of regulation, and epigenetics is the closest level to the cell, because to the genes, because that's actually the last kind of level of genetic control, because it's on the actual genes, on the DNA, or on the histones where the epigenetic regulators are sitting. Like methylation of DNA is actual control on the gene itself. And another evidence of this is that we have found that there are methylation clocks that ticks with age in our organism, in different tissues, that we can actually tell how old someone is based on their epigenetic profile of their cells, which is quite surprising, that there's actually some kind of synchronization between people of the same age, in different tissues, that you can look at their cells and say, okay, these cells come a 40-year-old or a 60-year-old person, which is pretty surprising. And from different tissues, even within the same organism, very different tissues show the same epigenetic time. The good news is that rejuvenation is possible, not just in nature and in the lab as well. Of course, in nature, we know it's possible because that's what happens every time a new baby is born. During fertilization, a cell is rejuvenated, an egg cell is rejuvenated, and it gets rid of all the accumulated damage that has accumulated within it until then. So an old egg cell, a human egg cell that's 20, 30 years old, becomes a completely new human without any accumulated damage. And of course, and the egg cell is 20 or 30 years old, just as old as the mother that carries it, because the eggs are generated during embryogenesis, the time when the mother herself was an embryo. That's the time when her egg cells became formed. So however old the mother is, the same time was passing to potentially accumulate damage in the egg cell. And it was actually shown in different papers that fertilized eggs can get rid of the damage. This was observed in nematodes, this was observed in mice, and I think it's pretty obvious this happens in humans when the egg is fertilized, that all the potentially accumulated damage is gotten rid of. Also in yeast, this was observed that yeast can not only reset their lifespan, their reproductive lifespan upon sporulation, which is a form of reproduction, sexual reproduction, but they can get rid of the accumulated damage as well, which was a bit surprising because they thought yeast just throws out the damage, but no, actual yeast cells, if they have accumulated damage until sporulation, during the sporulation process, they know how to get rid of it. And in nature, there are other ways we can observe rejuvenation. I'm sure you know about the jellyfish, the Turritopsis jellyfish, that is able to, from a old organism, become essentially an embryo of itself and rejuvenate the whole process, which probably is not applicable to humans because we want to keep our memories. But we know that potentially this is possible in an organism. And there's another whole genus of jellyfish, the Aurelia, that also know how to do this, basically turn from an adult to an embryo. Closer to humans, there are insects, beetles, that also know how to come back to previous stages of metamorphosis. For example, this beetle in the lab, this in such a way was, lifespan was increased by almost 10 times. This beetle was starved for food, and that made it return back to previous stages of metamorphosis. And that way the scientists were able to, kind of like the voles in Montana voles that were putting on pause their development, this was done in the beetle. They're rolling back their development, and this way, instead of living just for 8 weeks, this way they live for 2 years. So now we come to epigenetic rollback, the idea that you can use epigenetics to periodically rejuvenate the organism. And it all began, and the field of epigenetics began in probably 1940s. You can say that this is the moment that the field was started by Conrad Howell Waddington, the famous Waddington landscape, where he proposed this concept of epigenetic differentiation of a cell, how a cell becomes a stem cell, becomes some kind of differentiated cell, a skin cell or a brain cell. And he had this idea of a landscape, I'm sure you've heard of it, where it's like a stem cell rolling down the hill, and it, depending on the path it takes, ends up at the bottom being this one or another type of differentiated cell. And at the time, the thinking was that this is an irreversible process. Like a cell cannot climb back up the landscape to become a stem cell again. Once it's a differentiated cell, it cannot change its identity. And the thinking was that maybe during this kind of rolling down the hill, what happens is that certain genes are removed from a cell or dissolved in a cell, and that's why it only retains the genes responsible for it being the cell, the ultimate cell that it is. And I guess the inspiration for this was the red blood cells that, people observed that they actually don't have a nucleus. So they thought, maybe that's what happens in another cell. They get rid of some parts of the genes, and that's why things are irreversible. But that idea was reversed, or at least, the evidence contrary to that idea was provided by Sir John Gurdon in 1962 when he took a cell, a somatic cell from an adult organism, and used it in a previously enucleated egg cell to show that actually, all the genetic information necessary to make a new organism is still retained in the fully differentiated cell. So the idea that something is lost was incorrect. So there must be some other way that differentiation happens, and maybe that way is not irreversible. And this was proven to be true conclusively in '96 with the Dolly sheep that was able to be cloned, fully cloned from, again, a differentiated cell, where they took the DNA of that differentiated cell and put it in an egg cell of another sheep. And they actually, the sheep that was born was obviously from the DNA of the donor and not the egg cell, which, they actually used a very different type, very different looking type for the surrogate mother, so that they, once the lamb is born, they know for sure who it came from. Was the DNA from the donor, which looked like this, or was it from the mother, which looked like this? And once Dolly, was born, it was obvious that it actually was cloned. So the DNA that established its ultimate fate was the one that they inserted into the previously enucleated egg cell. And here's the one of the creators of Dolly with his creation. There are a lot of myths surrounding Dolly. Some people, for some reason, thought that Dolly was born old or she died prematurely because of some genetic aberrations. but those myths were quickly put to rest. I mean, Dolly did die prematurely, but she died from a viral infection. But there was nothing wrong with her DNA. They actually then cloned many more dollies. They're like at least 4 of these in this photo, and they lived a normal lifespan. And since then, this process of somatic cell nuclear transfer has been used in thousands of experiments, showing that there's nothing wrong with the DNA of the donor cell. And not, I mean, that's why we can reproduce these results. The biggest breakthrough, I think, in our field of epigenetic rejuvenation came in 2006 with Shinya Yamanaka's discovery that you can actually, like he completely refuted this idea of irreversibility of differentiation, this dogma that you're rolling down the hill and you can't get back up. Yamanaka showed that you can, in fact, get back up. A skin cell can retrace its steps back and become again a pluripotent, essentially an embryonic cell. And the genius of Yamanaka was that he looked at the embryonic cells, what kind of genes are expressed in them, and was able to find 4 out of a list of 24 different genes, the 4 key genes that are actually responsible for not only for keeping a pluripotent cell pluripotent, but also for being able to make an already differentiated cell become back a pluripotent cell, which was a very non-obvious thing. And that's, I guess, that's the biggest luck that we as humans have, that it actually turned out that this is very potentially simple process of just, if you are able to express the genes that are responsible for this pluripotency in a terminally differentiated cell, they actually turn themselves, they know how to return back the differentiated cell into this pluripotent or stem or embryonic state. And so they had to update the Waddington landscape or epigenetic landscape to be able to retrace the steps back. They made it reversible. And now we know that, it doesn't matter where you as a cell are, you can always end up, well, unless you lost the nucleus, like a red blood cell. But if you have the nucleus, you can return back to pluripotent state. And this led to the idea of people in our field starting to think, okay, can you then use this process to rejuvenate cells? Because it was observed that cells that are with the Yamanaka factors turned back into embryonic cell, pluripotent cells, they're actually rejuvenated. So the question was, can we split this rejuvenation from becoming again pluripotent? And from what I found, the first person to think about this, or at least to put a paper with the words epigenetic rejuvenation, was Prem Singh. And this is the 2010 publication in which he actually talks about, can we use this paradigm of epigenetic rejuvenation to, or the Yamanaka factors to have epigenetic rejuvenation. And then the next paper that I saw in terms of the timeline is a 2011 paper that actually observed this rejuvenation in the iPS cells, or the cells that have been using Yamanaka factors returned back into pluripotent state. And this was under the direction of Jean-Marc Lemaitre from the French Institute in CERN, where they showed that even centenarian cells, like cells from 100-year-old people, were fully rejuvenated. And this opened an even bigger door into the investigation. Okay, can we now use this power of Yamanaka factors to rejuvenate cells in the organism? And the other clues that we saw that this is possible, it was from other publications that have looked at various hallmarks of aging in the context of what happens to them during reprogramming. And they show that all of the hallmarks, cellular hallmarks of aging are rejuvenated in the process of using Yamanaka factors in reprogramming. And parabiosis was another observation that was another earlier clue that this could be potentially possible. Okay, I saw that I have 5 more minutes left, so I'll try to speed up a bit. Another important step in translating this paradigm was from Tom Randall, who actually asked the question already, like in the paper, essentially saying, okay, can we now use the Yamanaka factors in the context of rejuvenation in an organism. And this is the paper in 2012 in which he asked this question, and it inspired, or I think it inspired Manuel Serrano, who tried doing this in a mouse model. He created transgenic mouse models where you can induce Yamanaka factors on demand. And he, in that paper, he did not look into rejuvenation, but he saw that, you know, a 7-day course of reprogramming actually results in teratomas. So his paper was less about rejuvenation, more about what bad things happen if you induce Yamanaka factors in vivo. And a parallel discovery was that you can actually fully rejuvenate mitochondrial function with inducing these factors in vitro. If you take centenarian cells, you can restore mitochondrial function after reprogramming. But the biggest breakthrough, at least in my mind, was the 2016 paper from Alejandro Ocampo and under the group of Juan Carlos Espiso Belmonte from Salk, where he showed that using partial reprogramming, you can greatly extend lifespan in progeric mice. You can extend it up to 50% if you look at this control group, or 33% if you look at this control group. And I won't get into the details because of the time constraints, but the most important part is that the mice, they not only looked younger, the treatment group, but they were physically younger based on various parameters, you know, senescent cells, double-strand breaks, etc. And if you look at their tissues, they were much closer to wild-type tissues than to the control group tissues, which, you know, it's visually striking that, you know, the treated group was much closer to wild-type animals than to their animals of the same age that did not receive the treatment. And they had fewer double-strand breaks, DNA double-strand breaks, they had more hair follicles, and even wild-type mice after the treatment they showed rejuvenation in their tissues, in their muscle tissues, for example, on this slide. But the biggest problem is teratomas. And you know, to avoid teratomas, what we need to do is to limit the expression of Yamanaka factors either temporally or by tissue, to avoid tissues where the teratomas can happen, or to have a very short duration of Yamanaka factor activation so that teratomas just don't have enough time to form. And I'll skip the couple of articles that show what happens when we don't limit the expression of factors temporally, but just show that if you do limit this, for example, in the brain, only a 7-day duration of Yamanaka factors, there are no teratomas observed. But the greatest observation about partial reprogramming, why we think it's possible to have epigenetic rollbacks, is that reprogramming happens gradually. So the epigenetic age decrease during reprogramming happens gradually, and loss of identity by the cell also happens very gradually. So before a cell loses its identity, before it stops doing its function, it is rejuvenated. So we can find a therapeutic window where we can have both. We can have rejuvenation without loss of function. And this is what Prem Singh again observed in 2019 and wrote in his paper. I'll skip the epigenetic clock because I'm sure you know about it. Just tell you what we try to do as a therapy. The idea is to be able to freeze our mortality rate, which raises exponentially, to freeze it at least, you know, a 60-year level. Because even if you're 60-year-old, you think you're old, but your mortality rate is not that large. It's 1 in 100 annual mortality rate. So if we freeze it at that point, you can still expect to live 100 years. Of course, you know, our goal is more closer to this, but even if we, you know, reach this goal with the therapy, which periodic rollbacks will be, you know, at least stopping the exponential increase, that'll be good enough for us. And the idea to how the therapy would work is that you integrate the genes responsible for the reprogramming into the organism once, into the right tissues, and then you periodically activate them with a small molecule, just like in the Ocampo experiment where they use doxycycline to activate those genes on, they use the weekly periodicity. I think for humans it would be monthly or maybe even yearly because we have much slower processes happening. And this would what we hope to accomplish using the therapy at Youthereum. And the R&D goals, I mean, I'll skip over because I'm running out of time, although I thought I still have until the hour. But basically, our goal is to first make sure the therapy is safe, to avoid teratomas in fast dividing tissues, and then to also be able to activate those genes with an additional level of safety. So we want to build in safety logic to prevent overexpression of Yamanaka genes to avoid teratomas. And I'll skip the business part. Basically pets and cosmetic applications are the first two areas of focus. We want to have GMO pets and maybe extend lifespan in animals for existing pets as a first go-to-market product. But we're also considering other alternatives as well. And right now we're fundraising our $2.5 million round, which will focus on novel IP creation, as I mentioned, the cassette that will have safety logic built into it, and also on further proof of concept testing. Okay, I hope I still have some time left for questions. Although I thought I have until the hour, but maybe if I don't, please correct me if I'm wrong, Natalia.
We are already out of time, but still, thank you so much. And does anybody has any questions? Okay, we don't have questions. Do you want to say a couple more words about your own work, about the next project? Because it was very, very short.
Me? Okay.
Yeah,
When you told me I have 5 minutes, I thought. So I do have until how much time do I have?
No time at all, but still it's very interesting for us to know about your own project. It was very interesting introduction, scientific introduction, but we are also interested in what are you doing?
Sure. So we are, as Youthereum, we are translating this science of partial reprogramming into a therapy. And so the commercialization strategy is, as I mentioned, there are two different tracks. One is for pets, because we think, this is the easiest go-to-market strategy due to lower regulatory barriers and just, being able to much quicker use GMO pets because we avoid any kind of delivery issues that we need to do for gene therapy. So we can create, transgenic mice with this cassette built in. We can create transgenic pets like dogs or cats with a cassette built in, where the owners would be able to periodically activate it to keep their pets younger or extend their lifespan. But of course, our ultimate goal is a human therapy, and ultimately, that therapy would be able to, if it works, it will be able to prevent all sorts of age-related diseases. First and foremost, probably Alzheimer's, because this is a huge unmet clinical need, but also cancer and heart disease is the two biggest killers. That any rejuvenation therapy, if it works and if it reduces biological age, it by proxy reduces the chances of development of all of the age-related diseases that are associated with a given biological age. So the market for pets, I think it's very kind of self-explanatory. There are a huge amount of money people spend on their pets, including on, not just on food, but on also various treatments. And if there was a therapy that was able to prolong lifespan of people's pets, they'd be willing to spend money on it, even more than they're willing to spend money on themselves. Many pet owners are, spending more money on their pets than they spend on themselves. But the next very logical category is cosmetics, because it's a huge market in itself with very little actual efficacy. I mean, none of these therapies that claim to be rejuvenating or anti-aging are delivering on that promise. So if there was a therapy, even if it was a topical administration, like a skin administration of the Yamanaka factors that does actually rejuvenate on a physiological level the skin, that would be a very attractive product in itself. But ultimately, we want to have a therapy that integrates into many tissues and has a systemic rejuvenating effect. In terms of deliverables, as I said, pets are probably the quickest go-to-market product. After that, we want, GMO pets are the quickest because they don't have integration issues. But the next step is to have a therapy for existing pets, which use an AAV or something, or some kind of delivery into the existing organism and is able to rejuvenate the organism, and then also a sort of like a testing platform for humans. If it works in animals, the next step is having clinical trials in humans for some kind of age-related indication to prove safety and efficacy, and then enter the market with the human therapy. So yeah, very quickly, that was the our current outlook for the project. So about the round that we're raising, as I mentioned, it's $2.5 million to finish the very early preclinical proof of concept stage of the product and to narrow down the safety, the cassette with the built-in safety features that would be able to integrate into the animals to avoid overexpression of Yamanaka factors.
Okay, thank you so much.
Very much, Yuri. Thank you very much.
Thank you. My pleasure.